Sure to hit it on the cloud. So welcome everyone to our Wednesday webinar. We're rounding out the fall schedule. We do have one on December the 6th for HSEA members, and that will be at 7 p.m. Eastern. So just a heads up on that. We do have one more for HSEA members, but to round us out for the regular fall season, we are excited to have Damon and Tammy with us. They're going to talk to us about Biotechnology, a new HOSA competitive event, and BACE, the nationally recognized biotechnology certificate. They are from Bio-Rad Biotility. So welcome you guys for joining us. We're so excited, and we'll go ahead and get it turned over to Damon and Tammy.
All right. Thank you for having us. Really excited to be here and kind of share what's going on as biotechnology becomes a bigger portion of like health science education. I'll get things kickstarted by kind of going into the HOSA event, doing a little bit of background on biotech, and then I'll turn it over to Tammy. As I'm going, feel free to kind of pop questions in the chat, or when you see me giving a breath and you want to get a question in, feel free to unmute and jump in. We'll just work it out from there. I'm gonna go ahead and share my screen and jump into this presentation. Let me just make sure everybody can see that. Just give me a thumbs up if you can see that, Nancy. That way I just know everybody else can see it. Great. Okay, so my name is Damon Tighe. I work for Bio-Rad, which is a big biotech company that's been around since the 1950s. We kind of do support of everyone that's in the life science research world and in the clinical diagnostics world. So we're like somebody that sees a lot of how biotech gets its way into kind of the medical community. And so I'll be kind of hitting a couple things within this talk, mainly giving you all the details you need to kind of round out why there's this new HOSA event, what it's going to look like, and things like that. So students over the last couple of years have been exposed to a lot of biotech vocabulary through routes you probably would have never expected, which was through the general media due to the pandemic. So they've picked up on a lot of these terms, and they haven't always picked up on all of the content that goes along with that vocabulary. So this is a really interesting period of time because we've got a chance now to take a lot of these things that they at least can say and help them fill that out, understand them, understand the skill set, and then how that applies to the medical community at large. And so that's everything from words like PCR, the polymerase chain reaction, to real-time PCR, basically a way to monitor a PCR reaction in real time very quickly. Antigens, antibodies, all these things that were incorporated in our diagnostic processes for understanding the spread of the SARS-CoV-2 virus. And so I think there's a giant opportunity right now to build on this vocabulary by teaching them the theory and the skills. And this all feeds into a general market that is growing enormously fast. I mean, biotech's been growing very strongly for the last 20 years, but definitely in the last 10, it's taking these giant jumps that are on pace with what we've seen in the tech sector. And it's interesting because biotech touches a whole bunch of different kind of domains of industries, but it's always been a large part of what's going on in medicine, everywhere from the research to the diagnostics to the actual therapies. And so HOSA in the past year has launched this new kind of biotechnology competitive event. And so it was really interesting actually getting to go to a couple of these last year and help facilitate the competitive events. And so I'll dig into a little bit of that more at the end. Remember, this is kind of different though than kind of the kind of biomed. Biotechnology, right, is kind of the science behind a lot of the medicine. So it's more focused on those type of skills, that type of theory, and then also understanding how biotechnology makes the tools for the medical marketplace. So just to kind of give you a general foundation in what biotech is an ancient kind of domain. We've been more or less doing biotech ever since we started doing cultivation of different organisms to get us things we wanted. So that's things like making bread, or using that same organism, Saccharomyces, the baker's yeast, to brew other things that humans might want. I mean, you can find protocols for making this type of stuff that go back to 6000 BCE. You know, they weren't as streamlined and they didn't have the, CGMP and quality control groups that we have now in biotech, but they were definitely regimented protocols so that people could reproduce these processes. And what students are going to be exposed to with the HOSA biotech, though, is not beer brewing and bread making, but more of the contemporary biotechnology tool set. And so that's going to draw on their knowledge that they're probably picking up in a lot of classes already, which is genetics, molecular biology, some biochemistry, some cell biology, microbiology. And then once we get to the manufacturing side of biotech, which is a really big portion of it that isn't always highlighted in a lot of curriculum, you also have a lot of engineering, physics, chemistry, and computer science that all fits in there in order to be able to fill out this entire industry. So a great case study, actually, that I use a lot of times when I'm first talking to students about kind of contemporary biotech is the case study around insulin. And so the reason I start with this is because most of the students, right, have some knowledge about insulin or diabetics. In fact, I've been using this as like a flashpoint in conversation with students for probably the last 10 years I've been working for Bio-Rad. And sadly, every time I go into a classroom over the course of those 10 years, I feel like the percentage of students that can raise their hand when you ask, do you know somebody that's diabetic, has just gone up and up and up. So now when I go to the classroom and ask this, it's usually like 60 to 70% are raising their hands, which is great because this gives them a reason to want to understand this, right? They've got a personal connection to it. And so I'll usually play this game with them, which is where does insulin come from? And then let them try and figure out where it comes from what they know. And it's usually like the doctor's office, the pharmacy, and then you ask them, okay, where do they get it from? You play this game going up. And eventually, you know, unless you've got some students that have already kind of learned about this, they kind of run out of where it comes from. And this then gives me a chance to kind of step in and talk about insulin as a whole and kind of how it was first kind of brought into the medical community, which was by harvesting pancreases from slaughterhouses. So these are pictures from Eli Lilly out there in Indianapolis way back in the day when they would just go to the slaughterhouses, get those pig and beef pancreases, cow pancreases, and then basically grind them up, do chromatography in order to separate out the insulin. And that's the product that people were injecting. And this was good at the time, but then in the 70s, there became this huge issue that was taking place, which was if you looked at the number of diabetics and you looked at the number of slaughterhouse animals, diabetics were growing like this, slaughterhouse animals were only growing like this. So what's going to happen? You're going to have a giant market shortfall, which means the price on insulin is going to go up. And I love doing this with the students because you realize all of a sudden that all of them are economicists at their hearts. They all understand that. When you show them that little kind of like graph with your hands, they're like, price is going to go up. And then you can talk about what does this mean from a public health point of view. This is a really bad situation because if people are now having a hard time getting access to this medicine that they're using to deal with their pathology, bad things start happening. And I usually like to kind of dig a little bit deeper in there because when you ask them, their first thing is, well, people are going to die. And it's like, yes, but let's talk about how it happens because it's really ugly. And this is definitely something we want to keep from happening. So once we lay that down, then we talk about the solution that arrived through modern biotechnology manufacturing, which was basically this kind of workflow here. And this was more or less a collaboration between Eli Lilly, the old pancreas juicer, and South San Francisco's company called Genentech. So what they did was they took the human insulin gene, they moved it into a bacterial plasmid, and then took that plasmid and then moved it inside of, I jokingly call everyone's favorite butt bug, the E. coli organism, and then we're able to kind of grow this bacteria up and get it to produce human insulin. And then what they could do is they could harvest those bacteria, extract the protein, purify it, quantify it, and run it through all the quality control. And all of a sudden, you had something that was injectable. And this was big for a number of reasons. The first time, this is human insulin. Before, what people were injecting was one amino acid off, which meant that some people would get immunological responses to it. So A, now we've actually got human insulin. B, you're not growing slaughterhouse animals that take months. You can get this turned around in 2 weeks. And so the scalability goes up. And all of a sudden, we basically miss this giant public health crisis of the early 80s due to the number of diabetics growing faster than insulin production. So I think this is always a good kind of starting point for them just to kind of understand conceptually why this is such a big deal. So modern biotech is basically providing us solutions that nothing else could have. And that's why it's really exciting. And this, remember, this is 1978, 79. This is the infancy right here of modern biotech. What's happened since then is incredible. And we want students to be aware of that and to be excited. This is also a field that they could be involved in that could allow them to make big differences in other people's lives through medicine, but not necessarily having to be in the clinical environment. So let me give you a little bit of this, more of the stuff I showed to students, but it helps you kind of understand the excitement of what's coming. So we have these emerging futures. A lot of people are just starting to kind of hear in the news and stuff like that. So gene therapies have been, on the tip of people's tongues for like 20, 30 years, but it's really only, I'd say, in the last half a decade that we're seeing the prospect of them become realities. And so the most recognizable one of these is Victoria Gray's story. She's in the clinical one trial for a CRISPR-based gene therapy to solve her sickle cell disease state. And it works for her beautifully. And so right now, this is in, I think, a combined phase 2, phase 3 trial. And so we're looking at this potentially being a therapy that's on the market within the next decade, which is really exciting. And this isn't the tip of the iceberg. If you go into clinical trials right now and just type in the word CRISPR, you're going to find just a list of things coming through. And so we're seeing gene therapies finally pay off from all this talk we've heard about in the last couple of decades. Organ printing has also come online extremely fast in the last couple decades. So the first real big breakthrough was in 2001 when people were starting to print bladders and then be able to put them into people. And then you had things like livers, kidneys being done in animal models like pigs. None of these have been done in humans yet, but we're definitely edging our way there really, really quickly. External tissue stuff like dermatology stuff like ears and things like that, we've got that down really well now. We can print you basically a bio pattern of collagen, say of your ear, take your stem cells and then regrow that onto that surface and then implant those things. And so this is definitely a field that's kind of exploding really quickly. I always give this one to students because they get really excited because they're like, oh, cyborgs finally. And I remind them, they're like, actually, cyborgs have been here for a while. Most of your grandparents probably are a cyborg. Because since the 50s, we've had pacemakers, which is not as cyborg as you can get, right? You've got a mechanical device that is keeping you alive. Or even the 80s, where you have a mechanical device that listens to the world and directly talks to your neurons so that you can hear things. And you know what? If you just implant that with a different microphone, all of a sudden you've got superhuman hearing. So that is about as cyborg as it gets. But I know what students really think is like 2014, where this gentleman here is a double amputee, and APL basically replaced him with 2 robotic arms that can take basically nerve signals that are coming from him thinking about his old arms and then translate those into movements in his mechanical arms. And then in 2018, things got really weird because a lot of this kind of robotic-y stuff started going into biohacker spaces, where these are people not in academia, not in business, but kind of a lot of tattoo artists that have been used to putting stuff under the skin, deciding to put really weird stuff under the skin, like LEDs, RFID chips. Gentleman there on the right is colorblind, but he built himself a way to stimulate his brainstem so that he can see colors using this device in front of him. So it's a really weird, explosive world out there at the leading edge of kind of like biotech meets biohacker world. But all of this is to come back to, it's a really exciting time to be in biotechnology when it's applied to medicine. And that's both on the research side, which is understanding the fundamental biology of what's going on with diseases and health and human basically progression. The diagnostic side, which we've seen a lot of in the last couple of years due to SARS-CoV-2, and of course the therapeutics and prevention side. And so biotechnology really is that science behind the medicine. And for like someone like me who started off thinking they're going to the clinical science world in high school, this was the route I ended up going because I learned that I loved medicine, but I didn't like the clinical environment. And biotechnology allowed me to do what I really loved, which was the application of science for community good, but not necessarily in the clinical environment. And so this is also a big pathway for a lot of students that like medicine, but maybe clinical environments aren't for them. So there's a lot out there when it comes to like benefits from patient care. When it comes to biotechnology, there's a ton of career opportunities. Everything from actually being in a clinical environment where you're helping, say, medical researchers run experiments and tests for clinical trials, all the way through more of like the traditional kind of lab zone. And then even there's some places that are kind of emerging right now, which is people that understand genetics and bioinformatics and are being able to do kind of genetic biocounseling with folks as well. There's also been a big push from the recent US administration to really increase what's going on in biotechnology and biomanufacturing. So there's a lot of education money pouring in there, in addition to a lot of money allowing us just to grow the biotech and medical market when it comes to biotech applied to it. So let's dig into the HOSA competitive event now, since you've got a little bit of idea of what's going on in biotech. Actually, let me give a little breather in there. Are there any questions on any of that stuff before I move on to HOSA? I'm really bad about breathing sometimes. So I'm going to take a sip of my coffee. And if anybody has a question, you can get it out there right now. Okay, we'll go on to the HOSA competitive event. So let me give you a background on why Bio-Rad's involved in this, and then I'll give you like the more of the details on the HOSA event. So Bio-Rad got involved mainly because we're somebody that's really interested in growing the overall biotechnology market, and we realized that education is a big part of that. If you don't have people that are understanding these technologies, there's no way for kind of the markets to grow. And we're a company that's been there a long time and really helped the field grow in many, many different ways. The beauty about Bio-Rad is we're a very kind of long-term looking company. Like I rarely hear about them talk about things like as quarters. It's like, what are we doing over the next decade? How are we changing the world as far as being able to increase what's going on in the research and in the medicine side of the things that we do? So we're a worldwide company with almost 8,000 employees, almost 140 locations, and we operate in 35 different countries. So a lot of times people are like, oh, Bio-Rad, I know you guys, you're down the road from me. And it's like, well, there's a good chance of that because yeah, we've got a lot of locations. Most of our companies kind of split into two major groups. One basically works for life science product solutions. So this helps people on the research side of the medical world. These are primary, science folks that are trying to figure out how does this disease state come to be, things like that. And so we offer all the tools for people to study those types of things. And then on the other side of the company, we do clinical diagnostic work. So a lot of those kits that get used in hospitals are things that Bio-Rad are producing. So we produced like one of the first HIV kits in the world. We produced one of the first mad cow disease kits in the world. We do a lot of kind of bloodborne work and things like that. And then about 25 years ago, we had a high school teacher that joined us who was like, you know what, it'd be really nice if all the stuff that was going on in biotech, I could actually get in my classroom and not the way that I've been doing it, which was like dumpster diving to get stuff and then more or less building out curriculum until I ran out of that stuff. And so luckily, because that long-term perspective, the CEO at Bio-Rad gave Ron Mardigian some space and some time and a little bit of funding and said, what would you do to bring this into classrooms? And so what Ron did was he took kind of what Bio-Rad was building for researchers, then figured out a way to kind of take those same items and then make it so that it was something you could easily bring into a classroom and then teach those techniques. So since then, we've served about 1 million students, and we train probably about 2,000 teachers every year, hands-on at conferences, through webinars and things like that. So let's now take a deeper look at the HOSA event. So there's going to be like classic HOSA event. There's kind of two sides to it. There's a written test and then a hands-on skills portion. The written tests, people are usually not as spooked out about, right? Because taking on new information, relaying new information, helping students learn it, that's what we've been doing in education, right? That's our bread and butter. But people usually get all worked up about the hands-on skills because maybe these are things they haven't done before. And this is where we're really there to kind of help support people understand how to go about teaching this, what tricks and tips and things like that are needed. So the written test, 50 multiple choice questions. You've got about 60 minutes to do that. Most of this content can be covered in a lot of kind of biotechnology curriculums you see out there already. We actually produce a biotechnology textbook that is actually a great tool for doing this because it has all of this kind of content in there. And this content that you're going through for the written test, this lines you really up for what Tammy's going to talk to you about in a bit, which is the BACE certificate. And so the laboratory textbook, there's a student edition and a teacher supplement for advisors. But a lot of that content you'll find also in AP coursework. You'll find it in Project Lead the Way. Biotechcareers.org has a lot of the kind of content you'll find in the written portion of the test. Biotechnology, but that written stuff also kind of allows you to do a little bit of career exploration in there, which is always something you want to do anytime you're bringing in a new industry, right? Is allow them to understand what are all those different positions look like. Because I know for me being a high school kid going into healthcare, the only thing I was aware of was what they showed me on ER. So I was like, so you're a nurse or you're a doctor. Forget the other, 100 of, careers that are out there. So the textbook allows you to do a little bit of that. So round two is really the portion that people get worried about, and this is the hands-on skills side of it. And this is where kind of Bio-Rad is really there to kind of support people. And this is why we went around to as many of these competitive events we could this year and kind of help support facilitating those events for the first time. So these hands-on skills are ones that require minimal equipment. There's really no hazardous chemicals or compounds students are going to use. Skills can all be completed in under 20 minutes. The skills reflect techniques that are common to most biology classrooms at this point, especially anybody that has like an AP classroom. Most of these are things you're already going to be seeing in those AP classrooms. And then we try to position them within relevant real life scenarios and skills. In fact, I'm going to take the competitive events, the different 7 ones, and then show you actually how they kind of map to something like producing insulin in just a second. So these are the 7 different hands-on skills that they might be asked to do in the competitive event. One is just demonstrating the use of micropipettes and transfer pipettes. And remember, this is like the core skill of biotech is you gotta be able to move discrete amounts of liquid from here to there precisely. And then there's DNA electrophoresis, basically showing that you can run and separate DNA on a gel. There is the bacterial transformation. How do you take exogenous DNA from one organism, move it into another one, and get it to express it? There then is one on just the interpretation of a gel. How do you interpret the banding patterns as far as the size and things like that of DNA? There's one on doing restriction digestion, which is just enzymes being used to cut pieces of DNA in discrete locations. Then there is the Bradford protein quantification, which is just a way to easily quantify how much protein is in a sample. And then a qualitative ELISA. So ELISA is a technique that's used to detect the presence of a specific antigen. So if you think about like most of the at-home tests that people have been doing for SARS-CoV-2, most of them are based upon the same technologies as are found in ELISA. So antibodies interacting with antigens and giving you a chromatic output. If I were to try and map these onto, say, a workflow we talked about earlier, like the production of insulin, micropipettes are basically used at every kind of step within like production. A bacterial transformation is used at this step when we've taken the human insulin gene and then put it inside of a plasmid. We use bacterial transformation to basically drive that plasma inside the bacteria. We would have used the restriction digestion and actually to open up the plasmid and help put that human insulin in there. We would use the gel electrophoresis to confirm that that had been put in there before driving it into E. coli. And then once we'd gone through and purified the protein, we would use a Bradford assay to actually analyze how much of it did we recover. And we might use an ELISA just to confirm, or antibodies to confirm that that is the protein that we've isolated. And so all of these skills map into kind of corresponding usual production pathways within producing drugs and therapies that are used within medicine. To support all of this, Bio-Rad built out a whole bunch of different tools. And so we have like HOSA guidelines, which make it really easy to understand what's being asked of students during those competitive events. The biotechnology textbook is a great way to get all of the kind of background content that they need for the written test, and also it has the protocols that they would be using in the hands-on portion of the competitive event. Then we also have an entire YouTube list showing students all of those procedures, what they look like in practice. And so we've got 3 really good kind of pillars for them to lean on for that competitive event. Since we're running a little bit farther on time, let me just kind of jump through to this. So to get any of those resources, if you have your cell phone, you can scan that QR code, and that will get you to the resource page that we have for all of the HOSA competitive events. We'll also send out the slide decks that you have it at the end. And that competitive event really appeals to students that have like a familiarity with biotech, right? And a lot of them already have this from going to that HOSA competitive event this past year, because a lot of these students are coming from these combined pathways where they've been doing a little bit of biomedical that has a little bit of biotech in there. And so all the HOSA advisors doing is kind of filling out more of that portion of this that is not covered by kind of a classic biomedical. And then if you're already doing the HOSA biotech competitive event and you're getting that content for them, you're pretty much almost already ready to then take BACE, which is this fantastic certificate that kind of allows industry to go, oh, this person has these skill sets and is employable. And this is something that high school students can go after, I would say, as they've done basically the training for the HOSA competitive event. And so if you combine basically the HOSA competitive event with taking some of the Bio-Rad's textbook that allows them to kind of dig deeper into the manufacturing knowledge, like CGMP, QA, QC type of stuff, they basically have a full package then to kind of be ready to go and examine for the BACE. And so with that, I'm going to go ahead and stop my share and I'll turn it over to Tammy.
Awesome. Thank you, Damon. And thank you all for being here. I know after school it's already late and some of you are probably ready to go home and take a rest. So I will continue to follow on, but although quickly, are there any questions for Damon before I move on? Oh, okay, cool. All right. So, Damon just, it's so exciting to see the advances in biotech and really, how students can align what they're learning in biomedical science programs with a whole nother career path that they're really not frequently even aware of. So I'm going to talk a bit about the BACE, but to be honest, my real goal and focus is to really start talking about, what are these careers and how the content that's embedded in your curricula really is an important part in these careers. And so just real quickly, I am Tammy Mandrell. I'm from the University of Florida, and I direct a center that has been in place now for almost 20 years that's focused on developing workforce talent for the bioscience industries. And we do this by direct training to the industry. I come out of industry myself. We also do a lot of educator professional development, but of course, today we're here to talk about the BACE and the careers. And if you aren't already familiar with the BACE, real quickly, it's an industry recognized credential, and it's the outcome of an industry DACUM analysis. So these are what they consider the minimum competencies and skills that they're looking for when they're hiring candidates into their positions for hire. And that's maintained by a national advisory board. And so what I want to do is I'll show you what these competencies and skills are. But the idea behind this is that, when you hire somebody who's earned this credential, they've shown the aptitude for the work, they've shown the perseverance to pursue, right, a higher level credential for these jobs. And what it says to industry is that they can be hired and then be quickly trained on industry specific protocols. And so I think sometimes people maybe are a little apprehensive to either prepare or sit for the exam because they think, oh, I never worked with a bioreactor or large-scale purification equipment. But that's not the purpose of the BACE. It's really taking those foundational techniques, many of which you're using in your classrooms, and the competencies, and then applying them to learning these higher order things once on the job. So the board dictates the distribution of these competencies and skills, and you can see them outlined here. And again, I'll share my presentation, so this is all can be quickly at hand. And you'll see there's a big focus on those technical skills, and many of which, right, Damon's already described, for the HOSA competition and for these other applications. And then correlating with that on the knowledge exam portion or the competency portion is the industry wants their candidates to not only be able to perform the skill, but they want them to understand the science behind its application and why it would be used, right? And so, Damon described, the ELISA, right? Very common for pregnancy tests, COVID tests, but it's really using the antigen antibody conjugate to determine whether or not a protein is present. And so, right, just the basics of the mechanics behind these techniques that, and how they work. And so this is an outline of what's on the knowledge exam for the BACE. And again, I think most of this is going to be relatively familiar to anybody who's teaching Project Lead the Way's biomedical science program, or right, any other biomedical sciences. Even AP biology covers a lot of this content as well. The areas that are highlighted in green are where I see traditionally lacking in curricula. And this is content that is covered in the Bio-Rad Explorer textbook. And then we also have a number of resources on our website, which I'll get to a little bit as well. And then this is just an outline of the practical exam. So again, you see it's really what you find in most classrooms, and it's just your standard equipment, but if students understand, how to work with equipment, maybe keep an equipment log, the concepts of calibration, maintaining a piece of equipment, right? That's kind of putting it into the industry context. And so what I'm going to do real quick too is, normally I don't throw this in, but I'm going to throw it in because I think it's so important, is that if anybody is thinking of sitting for the BACE exam, I think one thing that people aren't typically familiar with is that we offer a number of free resources to help candidates prepare. And so I just want to show these to you real quick because we really see a difference in how students perform on the actual exam and then the amount of time they spend in our practice exams. So if a school registers to administer the exam to their students, or even if a student registers to take it as an individual, they have access to all these resources for no cost. And there's no cost to register the site, right, to test either. And we have these two versions of the practice exams. You see A and B, and they're broken out into the same topics that I just showed you earlier in the pie chart. And so the students can go through these, teachers can monitor student progress. You can see where there might be a gap and to supplement it. And so these are really valuable tools. And in addition to that, we have other resources to help supplement, right, where we see those traditional gaps. But if a student or a candidate passes the exam, then they're awarded a digital credential, which they can then use, to put on their resumes, LinkedIn accounts, etc. And then the positions that they're hired into, and it's a full spectrum. And I'm going to talk a little bit more about these because Damon really laid the groundwork for this. But they can be hired into a whole variety of these positions. And we've been focusing on the biopharmaceuticals and the biomanufacturing piece, but a lot of this is applicable to other sectors of the bioscience industry. Ooh, what did I just do? Can you still see my screen? Oh, there we go. Right. So, our focus here today is, the healthcare and the biotech. But what I love about these programs is, again, right, these skills that your students are learning are really applicable to the different industry sectors. So it really opens the doors to a lot of career opportunities. And this is just an example. This is a list of companies that have hired BACE credential earners. And you can see it really spans that variety of the industry sectors. So we have Boehringer Ingelheim, biopharmaceutical manufacturing, gene therapy manufacturing. But then there's also food and juice companies, or medical device companies, agricultural-based companies. Yeah, and so just again, I think what excites me most is this opportunity for expanding these options for careers. And the one thing about the industry that I really like is once you're in the door and you show the aptitude for the work and the willingness to work beyond your job description and to learn more, they're going to grow you in the company. There's much less of a glass ceiling. So it's not so focused on the degree, right? Bachelor's, master's, graduate degree, as it is your desire and ability to grow within the company. And so I've seen people come in at the entry level, high school or the 2-year degree programs, and really rise into managerial type roles just by, understanding the processes and working their way up. I'm going to talk a little bit more about these different positions, and I want to talk about how, what you're teaching and even the BACE, how that aligns with these careers. Again, because these are really meaningful, high growth, rewarding career paths. And, my goal one of these days, get up on my soapbox here, is I want these to be deliberate career choices for students. Very often they enter the industry careers because they didn't get into medical school or graduate school, or their plan A didn't work out. And so by default, they say, oh, I guess I'm going to industry. And like I said, these are really great opportunities. And I would love to see this being a deliberate career path. So research and development in the industry. So this is where I find students, if I ask them, if they express interest in the industry, this is kind of the default position because it's most familiar to them. But this is where they're really looking, to build that next blockbuster product. So not just discovering it. A lot of times they license that from academic settings or research institutes, but taking that idea and then figuring out what you need to make happen to develop it and scale it up into a full product. And so, in the example of insulin, a lot of times we're using cell systems to make these products. We have to optimize those systems. What's the best media? What's the best growth parameters? What other things do we need to do so we get the highest yield? And then also thinking we can do this at bench level, but how do we translate that into huge bioreactor, levels? Because that becomes a whole different process there. So research and development, I think, is a really exciting place to be. The other side of research and development is what we call characterizing the properties of the product, and then assay development. I really wish someone had told me about this, early on when I love science, because, if you think about the product label and all the information that's on it, all those parameters need to be defined before the FDA is going to approve it for sale, because these become the product specifications. How pure can we make it? How stable can we make it? What's its shelf life? Are there any safety concerns? All this information is critical, and it's actually the benchmark that we use to determine whether or not a product that has been made, in a GMP facility can be released for sale. So a lot of work goes into identifying this. And then the other piece is, how do we test it? How do we test if our product is pure? Well, you're doing that in your classes. If you're doing gel electrophoresis, if you're doing PCR, we have all these different assays in place that help us determine the purity of a product. How do we know that it's doing what we claim it's going to do? Assays, again, these, the ELISAs, the immunoassays, the protein, concentration, Again, these are all things that are used to test these products. So once a product has been approved by the FDA, and then we're making that product, we have to test it before we can release it for sale. And so that's where the quality control unit comes in, is they take those assays that were developed in research and development, and now they use them. Again, this is like a job where I think, I wish I'd known of this sooner, because it's hands-on. You're doing all these techniques. It's very applied. It's rewarding. You're testing a product that's going to impact a human life. And once again, these are all techniques that students are learning in biomedical science programs. And then manufacturing. So, these are the people that make the product. And, it's really going to differ based on what the product is, what the process is for that particular company. But it could be working with bioreactors, large-scale purification systems. It could be as simple as being involved in buffer prep, media prep. But we have this whole system. Typically, we call it upstream and downstream processing. Upstream processing is where you're working with the cells, growing up the cells, monitoring the cell growth, harvesting the cells. And then downstream processing is when we actually want to now purify our product away from all that cellular debris. So Damon talked about harvesting or purifying insulin away from, that mush of pancreas. And you think, how in the world could they purify protein, a pure protein away from all that goo? And it's simpler now that we're using the cell systems, but you still need to be able to purify the product away from that cellular debris. So again, it's an exciting place to be. And again, techniques and skills that your students are learning. Now, the quality assurance group, this is a good place to be for students who like science, but don't necessarily want to do science. The QA group are the people that really oversee the regulatory compliance. So when you're making a product such as insulin that's going to impact a human life, we have to do that in compliance with the FDA and what we call good manufacturing practices. And this is really a culture of working. It's a way of working where everything is done in what we call a state of control. Because if we do it right in this manner, there's much less likely for something to go wrong. And so really the emphasis of working in these work environments is maintaining what we say is control. And so QA really oversees these operations to make sure that's the case. And a lot of times, well, it's done through documentation, making sure people are properly trained, conducting audits, and then reviewing all the data associated with manufacturing and testing these products before you make the determination of whether or not that product can be released for sale. So in QA, what they want are people who understand the science, and then can make informed decisions about quality parameters. So that was quick. I know I'm going through this quickly, and certainly, either after today's session, if you want to just touch base with me, I'm really happy to talk more. But again, the take-home message is that these are really good careers and they do make a difference. And I know a lot of students go into the biomedical sciences because they care. They want to have positive impacts on humanity, the world, right? Individuals' lives. But like Damon mentioned, and I've heard many times in the past too, is, sometimes students get into a biomedical science program and realize, these clinical roles maybe just aren't for them. And these are really opportunities then to really take what they know into a different direction and still have that impact on life. So before I move on, are there any questions? I know I'm going really fast. Oh, hopefully I didn't lose everybody. You all still here?
There was a question for you. It talked about, do high school students take this? Let's see. It was from Sheila. Do high school students take the exam and are they successful?
Yeah, it's a great question. So the answer is absolutely. As a matter of fact, the primary candidates that sit for the exam are students who are high school students who are in rigorous biotech programs or Project Lead the Way's biomedical science program. And a gap analysis was done. Project Lead the Way did one several years ago, but they're actually updating it because they also have now revamped HBS. And so if these students are preparing for the exam, like I said, I highly recommend making use of the practice exams, primarily because of those items I had highlighted in green, right? Understanding why do we have good manufacturing practices? What's the role of the FDA? What's the role of the quality control unit, the quality assurance unit, right? Understanding these different parameters, that industry culture, because it's really a paradigm shift, right? We all learn science in the context of academic research. How do you now use those same competencies and skills to discover, manufacture, and test a product? And then in terms of success, so the pass rate for high school students has been right around 50%. It's a difficult exam, and that difficulty level is set by our National Advisory Board. Again, because we are making products that are regulated and impact human life, the industry really wants to make sure that if they're hiring somebody based on a credential, that it's a credential of value. And so that's also why we maintain the ISO 17024 compliance. In terms of that pass rate, and I don't have the data I can show you, but where we see candidates struggling the most is with the applied math. And so if this is something that you're exploring, then I highly encourage students to just practice the math. How do you make a molar solution? How do you make serial dilutions? How do you make standard dilutions, weight volume measurements? And I think that is going to make a difference in the pass rate also. But I definitely, so the other thing with students I know in, these programs, and especially the programs you're teaching, is that they're all looking at post-secondary education. Or many of them are, right? They're not necessarily thinking they want to be employed directly out of high school, although of course life happens, right? So it's always a great opportunity. But credential earners are also snapped up in post-secondary scenarios, either as undergraduate research assistants or getting internship positions, or possibly working part-time, in a company as they're progressing through their post-secondary studies. So absolutely, secondary, yes, love them. Come join us. Damon mentioned this also, and I know Nancy had put this in the chat. I highly encourage you also to look at the biotechcareers.org website. This has a lot of information about these careers all over the world, as well as programs that support the careers. So it's really a great resource for teachers and for students. And then, of course, right, the Bio-Rad textbook. So we're super excited about this synergy that we have with Bio-Rad. This has been, years in partnership, and just a fantastic combination of opportunities to practice the hands-on applications, put it in the context of the theory, and then that translates to, right, success on the BACE. Well, I can't promise that, but it helps. And then I also want to mention that we do professional development for educators. I really think that it is helpful, if you're teaching that, if you're teaching some of these topics, if you're teaching an ELISA, for example, to put it in the context of an industry application, right, all of these techniques. And so IBTE, it's the Industrial Biotechnology Teacher Experience, and it's really designed to help educators do that paradigm shift from teaching science as an academic endeavor, or maybe as a clinical application, to really learning about what it's like to work in the industry culture, the industry lingo, so that they can carry that forward into their classroom and really help expand awareness of these opportunities to their students. And the BACE, it originally launched out of Florida in 2012. It's now pretty much used all over the country. It was featured in this year's Coalition of State Biosciences. Bio is the international industry organization for the bioscience industries. And so for them to now be looking at credentials and valuing credentials is a huge step. If you don't see your state highlighted here, it doesn't mean you can't use it. You can help pioneer it in your state. We are happy to work with you on that. I also want to mention that the National Science Foundation has funded expanded awareness of the BACE credential. And this is a really fun little video. I think it would be very informative to your students. It really showcases industry talking about the BACE, the careers, as well as credential learners, and why they sat for the BACE and the value to them. And also expands career awareness as well. I know I'm getting close on time. My intent wasn't, well, I'll leave time for questions. So last year we piloted the BACE as a HOSA competitive event, and really excited about this because, as Damon mentioned, students can be preparing and participating in these activities with the Bio-Rad experiences, but there's also the option, and I want to say and, right, that maybe this year your students participate in the Bio-Rad events, and through that gain the confidence to move on to sit for the BACE. They can certainly do that. It's available as a computer-based option. This year at HOSA, we're looking into offering it as the full paper and pencil practical format. And then if you want to register, this is an opportunity. And then if you want to speak to anybody on our staff about what it takes to become a test site, there's no cost for that. Again, it gives you access to all the resources, or about taking the exam. Please reach out. This link here will connect you, or come to our website, email me. But we're really happy to help however we can. Again, at the end of the day, my goal is to really expand awareness of these opportunities and help students and help you, make this happen. So with that, I will stop sharing because I know we're coming up on time. And if there are any questions for Damon or myself, we're happy to answer them.
Tammy, I think one question, I believe Sheila may have asked this. She was wondering about the cost for the exam, and if I missed it, I apologize.
Oh, no. Yeah, no, because again, the point of my presentation wasn't to, do a deep dive into the BACE. So the cost, it's $185 per candidate. That includes, all the resources, but it also includes a free retake. And that's new this year. So historically, the exam was $150, but then students had to pay $50 to retake the exam. And we really want to encourage students, it is a difficult exam, to take a retake. It also makes it easier on the teachers and the schools to not have to do a second invoice. If you are in a state where the BACE is on their list of approved industry recognized credentials, even if not, right, a lot of times it's covered by the Perkins funds, under CTE. And so that's also something else to explore.
She has a follow-up question of how many times can they take it?
Yeah, great question. So they can take the exam 3 times within the what is it? The academic year, right? So, but it also, so if you take the exam, say in April, right, your first attempt, you can now do a free second attempt, and then you can take a third attempt if you like. That all has to happen before December 31st of that year. But it's a 20-day waiting period between retakes also.
And we hope they're studying in those 20 days. Any other questions from our people on the call today?
Could I ask one question? I work in an online school. Is there any idea to offer this in an online version?
So it actually is available as a computer-based exam. A quick background on that. Historically, it was a written knowledge and then a hands-on practical. During COVID, our national board, agreed that we could move the full exam into a computer-based format. And then during those 2 years, there was a third-party psychometric analysis study done that showed very tight correlation between the outcomes of the 2 versions. And so the board voted last summer to make the computer-based a permanent option. So that's there. So what I like about that is, there's definitely pros and cons to both. But I do like that it expands access to, earning the credential.
Mm-hmm. So how would there was a large emphasis on the actual skills. How would that work then? How do you?
So in your
technical skills.
So in your course, is there any opportunity for the students to do any hands-on type work?
We are a purely online school.
Purely online.
We do simulations for other courses, like they do medical assistant. It's purely online, but through simulations.
And so do they do simulations for micropipetting or?
Not in that. I was wondering if that exists. That's the first time I'm looking into the biotech.
Oh, I see.
Mm-hmm.
Ooh,
not just for the exam, but for the actual learning for it.
Gotcha. There are some resources out there. I don't know, Damon, maybe you can speak to that a little more.
I mean, there's a couple of simulated modules you can do online. I'd have to send you an email probably with the ones that I found. They're ones that kind of developed really quickly during the pandemic, so I don't have them off the top of my head. But that's predominantly what I've seen in that space. And so they're kind of like animations, right, where students are moving pipettes around and then going through the protocols of how you'd use it. But it's hard, like you're getting general workflows, but I feel like you're not necessarily getting that tangible skill sometimes with them. So, but I mean, you see that with also medical stuff as you're doing that, right? So it's like hard to teach phlebotomy through a computer.
But we do it online. It's accepted.
But you do it. Yeah. So it's the same.
There's a clinical, specifically for phlebotomy, there is a clinical part to it. They have to find a clinical space and do it, though. Yeah, that's right. Okay. The whole theme is interesting.
Where's the school, Astrid?
Good question.
In Oregon.
In
And Oregon
Oregon.
just accepted the BACE credential.
Okay, you
And
want
that's
to
my background, so I'm interested in it. I just wonder how we could realize it.
Well, thank you for being here. You want to put your email in there, and that way Damon can share more knowledge with you because he's the smartest person on the call on the webinar. We
Okay.
decided that earlier. No,
All
and
right.
Tammy is tied with him for sure.
It's fine. You can
Okay.
give it all to Damon.
Oh, Yolanda, you are too. We are so appreciative of you all being here and providing this webinar for us. We are pleased that both Biotility and Bio-Rad are members of the consortium, so they have close relationships with health science state leaders across the nation and are interested in what you need for your students to be successful in the classroom. So with that, there's her email address at
Got it.
I'm trying to say her name right. Astrid.
Astrid.
Yeah. And then we will get out certificates of participation. So for all of you who were here live, you will get 1 hour of professional development. A certificate will be emailed to you by the end of the month. And we hope you'll plan to join us on December the 6th at 7 o'clock. Even if you aren't a member of the teacher organization, just come and learn more about it. And if you aren't a member, maybe you'll want to become a member after hearing about all the support that we can provide you. December the 6th, a Wednesday, you can register for that on our website. Thank you again, Damon. Thank you again, Tammy. We look forward to maybe seeing you both or at least a Bio-Rad rep and Biotility rep at our board meeting in January.
Sounds good. Thank you for having me.
Okay.
Thank you.
Thank you.